My guest today is Dr. Hanna Stevens. Dr. Stevens received an MD and PhD in neuroscience from the University of Illinois. Dr. Hanna Stevens leads the Psychiatry and Early Neurobiological Development Lab at the University of Iowa, Carver College of Medicine. Dr. Stevens is a distinguished professor of psychiatry and holder of the Ida P. Haller Chair. Her work leads groundbreaking research into the molecular and cellular foundations of early brain development and is pivotal in linking prenatal stress, environmental factors, and genetic influences on childhood behavior and the emergence of disorders. With her focus on the critical periods of pregnancy and early childhood, her research aims to revolutionize the diagnosis and treatment of conditions throughout the lifespan. In addition, Dr. Stevens recently published a review paper in Biological Psychiatry titled Developmental Disruptions of the Dorsal Striatum in Autism Spectrum Disorder. Now, this is an interesting area in the so-called basal ganglia, an area known for its roles in various learning methods, cognitive functions, and motor movements. The dorsal striatum receives inputs for the basal ganglia that in turn recruits various other subcortical regions used to orchestrate downstream behaviors. For those roles, this is a region of interest for autism. And now, my conversation with Dr. Hanna Stevens. Dr. Stevens, can you start with explaining to me about your medical journey and your background?
Absolutely. Well, let me tell a little bit about my interest in the brain and neuroscience, and that includes how I was trained medically as well. So when I went to college, I became fascinated with the idea that all our behaviors have these correlates underneath that occur in our brain. Chemical signals and electrical signals have the capacity to help us think certain things or move in certain ways and make choices, which was fascinating to me. And I just couldn't turn away at that point. So I decided I wanted to learn more about the brain, and I went on to do graduate studies in the brain. But at the same time, I wanted to be able to apply what I was learning to directly help people. And I was fortunate to get into a program where you can train in science with a PhD and as a physician with an MD at the University of Illinois. And I studied there specific ways that the auditory system helps us understand language and the auditory part of the brain, essentially. And at the same time, I began to be trained as a physician and a doctor in training. And I began to see that how people develop through their early part of their life is fascinating from just an experiential and how it affects people's lives perspective. And at the same time, how the brain develops was also beginning to excite me as well, and thinking about that. So I wanted to, when I completed my training there, I wanted to go into a field where I could look at development of people's lives and people's brains. And for me, that was training to become a psychiatrist, and specifically a child psychiatrist. And at the same time, then continuing to do research in an area that was really relevant to psychiatry and child psychiatry. And that because there were so many unknowns in child psychiatry about what led people to have the problems they were having, what led children to have emotional or behavioral problems, I wanted to start early and look very early at formation of the brain and think about how that might set up the brain to not work as well for a person. And so I began to look at prenatal or embryonic brain development. And then I've taken that from there, studying both how particular genes and differences in genes affect prenatal and embryonic brain development, but also how different environmental experiences or exposures in the prenatal period can change brain development. That has brought us here to this time when I've actually, way back then, decided I really wanted to understand the brain. I've now reached the point where I have gone beyond just thinking of the brain, which is already complex enough, to understanding that prenatal brain development involves interaction of the brain with a lot of other tissues, and in particular with the placenta that supports the development of the entire fetus or embryo, and we think is worth studying for understanding exactly how it influences the development of the brain. That's not something now that I do as I practice as a physician. I practice as a child psychiatrist. It's certainly not something I do in my everyday life as a physician. I don't think about the placenta when I'm trying to help children and adolescents with emotional or behavioral problems. But when I try to think about how we might prevent those problems, that's where the science gets me excited, to try to think about if we understand the biology that's happening as the brain is forming prenatally, then maybe we can prevent some of the problems for the kids that I see.
I kind of like to rate things. I think I've constantly rate things, and I think for me personally, the number one thing with autism research ought to be the cause. And there's so much that says the placenta or the embryo is where we need to direct more attention. So I was just so fascinating with knowing that you have a history with the placenta. Um, what is the placenta's role on the embryo?
That's a great question and a great place to start. So the placenta is really that intermediate structure that is there, that mediates things being provided by the maternal environment for an embryo, and that embryo growing and putting together its brain and, of course, all of its other parts that it have to grow. So the placenta provides nutrients by actively transporting nutrients from a mother, the proteins, the carbohydrates, the sugars that a mother consumes are actively transported to the fetus, so it has the building blocks to actually start to grow and put together its cells. And it also provides a lot of guiding molecules that tell it in part how to put those things together, but that's only in combination with what's already happening in the fetus itself. The fetus has kind of its own blueprint, and some molecules coming from the placenta can help guide it. Things like hormones, things like growth factors. And then the placenta has a big role in modulating the provision of oxygen, because all human life needs oxygen. And so oxygen exchange is very important in the placenta. And then the removal of things you don't need. So as anything grows, anything functions, it produces waste. All the cells of the developing fetus produce waste, and those have to be eliminated through the placenta in part as well. The placenta also seems to have become developed to limit the exposure of the fetus to things that wouldn't be good for its growth. And there's interesting active transport mechanisms in the placenta to keep out foreign type molecules, things that aren't natural in the human milieu, maybe poisons that a person, like if we're thinking of way back during evolution, a person might consume things that are considered poisons from plants or something like that. And today, chemicals or medications can be actively kept from the fetus by the placenta. So that's another very interesting role it plays.
Okay. So explain to me how the placenta from mother to mother could that be different? because I'm very fascinated recently over the last maybe 3 months about even pre-pregnancy, the mother preparing for pregnancy.
Absolutely. So the placenta is actually made up in part of mother's tissue. Part of the uterus actually changes after an embryo implants and becomes part of the uterus. So part of the placenta. So part of the placenta is of maternal origin, and then part of it also arises actually from the embryonic cells of the embryo initially. And so it has unique qualities that neither the mother nor the offspring have in some ways, because it's both of their tissues working together. So it actually is, it's not just that it varies mother to mother, but every time a mother has a new offspring, or different offspring, that placenta will be different that time. But there are key elements that seem to co-occur. So, for example, a problem in pregnancy that can happen to some people is that they'll develop a pregnancy-specific disease called preeclampsia, which makes the mother have high blood pressure and causes some problems with growth for the fetus. And the placenta is thought to play a major role in that. A mother will be at risk if she has preeclampsia the first time she has a pregnancy. She'll be at risk the next time she has a pregnancy. So there's some things that are kind of the same as the first time, but she isn't guaranteed to always have preeclampsia the second time. And so there are things that are the same and things that are different.
Uh, are you familiar with neurofactor? NK, no, NFKB?
Oh, sure. Yes.
Okay, could you tell me a little bit about that? Because I planned on doing a solo episode, and
Oh,
that might have been a large part of it. But if you can share, then that's data directly to me instead of my interpretation.
well,
So
yeah. What got you interested in NFKB?
just the roles of this time or this little epoch that we're talking about, and the roles with like DHA and omega 3, kind of maybe rescuing that. So it was just I just can't get enough about this time frame, and knowing or trying to understand when does autism actually develop on that timeline from conception to birth.
Yeah. So, I mean, that's a really, like, we can step out and take that big picture view first before we go into the details of the molecule. But the big picture view being that so far, everything that people have observed about autism is that as early as we can know what to look for, we're seeing the differences of autism in people who have that. Suggesting that, people used to think, well, is it a problem that occurs because of something that happens in early childhood? And people are becoming more and more convinced that it actually occurs before early childhood, during prenatal development. And some of the genes that have been associated with autism have pointed in that direction because they are genes that matter a lot for the development of the brain prenatally. Not to say they have no role at other times also, but that they're just so critical at those early time points. It seems like the weight of the evidence would just suggest it's happening before someone is born, and then we start to pick up signs of it as a child develops further and further skills, or they don't develop those skills. Sometimes that's what we see. So getting at this particular molecule. So NF-kappa beta is a molecule that plays a big role in lots of different processes inside cells of all kinds. So it plays a role in cells of the developing brain. but it also plays a role of cells of the placenta, and in fact seems to be one of the major factors by which placental development itself is regulated. So as the placenta develops, it's a tissue that appears for this limited period of time, the period of pregnancy, and it has to develop quickly and build itself quickly, and then it's done. And so it uses molecules like NF-kappa beta to form blood vessels and re-engineer tissue around those blood vessels, and monitor the right level of sort of tissue creation and tissue. Part of development of any kind is tissue destruction, because you have to have the right structure, and sometimes that means sort of chipping away, if you will, kind of like on a sculpture or something like that. So NF-kappa beta, through its major regulation of internal cellular processes, is really important for all of those structures forming in the placenta. And I can't remember, has NF-kappa beta been suggested to be an autism-linked gene? Is that one of the reasons you're interested in it, Ryan?
I'm pretty confident in saying yes, but I do have a tendency of, because of, I don't want to make this sound bad, but sometimes we're faced with the different silos. So this discipline can figure out something, but this one, it doesn't quite conceptualize that or integrate it. So it might have been from that, but I think data does show that it is involved.
I think, yeah, it rings a bell for me that it is. And I think one of the interesting things for us to start thinking about, a lot of people, as we just talked about, have been looking at autism genes for when do they play a role in brain development? And they've pointed towards all these prenatal phases of brain development. And one of the interesting things, since we're on the topic of the placenta, that's starting to be discussed is, what is the role of these genes, not just in the brain? We think of autism as a brain disorder, but also in structures that support brain development, like the placenta. So we just discussed how NF-kappa beta has a very important role in the placenta. And so one hypothesis that is an interesting one that some people are starting to explore is the risk for autism-like problems arising not just from a role of a risk gene in the brain, but in combination with the fact that that risk gene also affects the placenta. The two things happening together. We've been doing some work not on NF-kappa beta, but on another autism risk gene, or genetic risk, which is a copy number variant called 16p11. Have you heard of that, Brian?
I've heard of 16p, yeah.
11.
1.2?
Yeah,
11.2?
exactly. Yep, that's right.
Yeah.
Yep. So that's either a deletion of a part of a chromosome or a duplication, and it's associated with a higher risk of having autism or other neurodevelopmental problems. And we have worked with a group at the University of Iowa that has a mouse model of that copy number variation, and have found that the placenta of that mouse model is not like a typical placenta. It seems structurally different, and it seems to be functionally different, like it's sending different hormones and growth factors to the fetus. And one of the most interesting things is that the placenta in general is really different in males and females. And it seems that this risk gene, at least in this particular instance, seems to affect male placenta more than it affects female placenta. And so in 16p11, it affects 16p11.2 affects both boys and girls. They both have a risk for neurodevelopmental disorders if they have that genetic problem. But there are some problems that arise in that disorder that are more severe for boys. And so this is one possible source of that difference between boys and girls that might arise from the placenta being different in boys and girls.
Oh, wow. That's very fascinating stuff.
Oh, yes. It keeps us, every new discovery just makes us ask even more questions.
Yeah, it just provides information about you guys have no idea what's happening.
Sometimes
There's
it
so
feels
much
that
more.
way.
Yeah, yeah. And that wasn't a shot against you. I'm just saying
I
overall, it's
agree.
like, oh, okay, there's this now.
Yeah.
You mentioned oxygen earlier, and also I wanted to talk about the tissues too, because something else that's I'm always fascinated with because if it's autism, there's autism and I call it XYZ. There's so many comorbids going on. With the tissue, something that whenever I saw this data, I was just like, oh my gosh, autism and leukocytes. It's like, what is the connection there? And as you describe the tissues and so forth, I'm just wondering if you have any insight into that. If not, then that's fine too.
We could probably talk a lot about that. I'm wondering, are you thinking about leukocytes during prenatal brain development, or are you thinking about leukocytes in general?
I was thinking in general, but everything that I as as I discover or come across, I bias towards this time frame that we're talking about.
Yes. Well, it's interesting because one of the things you might know about pregnancy and prenatal development is that because the tissue of the fetus is different than the tissue of the mother, the immune system of the mother could have a reaction against the fetus. And one of the major things that sustains pregnancies is that there's an immune shift during pregnancy so that there isn't rejection of the fetus. And obviously, leukocytes are white blood cells, which are a part of the immune system and allow us to fight disease and clean up stuff that's not supposed to be there. And they do seem to function differently in pregnancy, and they do populate the placenta. And they populate the placenta both from the maternal side. So mom has white blood cells that play a role in placenta and regulating the functions of the placenta. And then the fetal white blood cells also start to develop. They're not there right at the beginning, but they start to develop and also will play a role on the fetal side of the placenta. And immune perturbations during pregnancy are thought to be one risk factor for neurodevelopmental disorders, including autism spectrum disorder. So I think leukocytes, we don't know enough yet, really, about all the ways they could be altering placental function, altering brain function. But certainly the idea that inflammation is inducing a risk does mean that leukocytes have a role that we need to understand better.
Okay. All right.
And, leukocytes are also a very interesting sort of window into the biology of a person because there's something that we can actually look at from the blood of a person. We can't like take brain samples from people. That just doesn't, we can't do that. But we can take leukocytes, and leukocytes are thought to maybe sometimes have biology that allows us to understand other things that are happening in a person's body. But there are limits to that because leukocytes are white blood cells and brain cells are really, really different than white blood cells. So,
Okay. Do you know much about the brain development and things like cortisol or serotonin from the mother to the fetus?
oh, yeah, those are two very interesting molecules during pregnancy for sure. Should we talk about those? Yeah.
Yeah, the serotonin one, it really captivates me because of how its role and where it comes from, tryptophan and the melatonin and serotonin right there in the biosynthesis, and just serotonin's role in developing like the sensory map of in the central nervous system. So
Yes.
yeah, Yeah, let's get into that.
Yeah, absolutely. So, as you've already pointed out, serotonin plays a big role in putting together a brain that can detect stimuli that matter, like sensory information. Things like, a map of like the where on the skin, you might feel a stimulus or something like that. Um, and serotonin prenatally, I mean, so in general, serotonin comes from parts of an individual's own brain, and can affect our, like, within my brain I produce serotonin and it affects my brain development. But there's a stage of development during prenatal life that the serotonin comes from the placenta. So this is actually one of the best explored aspects of how placenta and brain are connected in their development, in that people have actually manipulated in mouse models where we can understand this biology better, placenta specifically in the serotonin specifically in the placenta, and shown that that basically completely changes how the serotonin system develops in the offspring, because it relies on that serotonin from the placenta. And so some of the idea behind the importance of the placenta for brain development is focused on babies who are born too early. And then while we've become better and better at allowing and finding ways to support those babies to help them survive after being born early, one of the things we know is that we're missing a lot of the factors that the placenta produces, while we are supporting their survival, supporting their development in many very, very important and positive ways. There are theories that missing things like the source of serotonin in the placenta is affecting the brain development of these prematurely born babies, which is one reason why neurodevelopmental disorders may be more common in babies who are born prematurely. It isn't just that they had a lot of stress, because they do have a lot of stress, certainly, because they have to be cared for so carefully, and there's a lot of risks to them. But it may be that we're lacking some of the key elements like serotonin coming from the placenta. And there's another molecule, and I know you had an interest in also in oh my gosh, I've blanked. What was the other one you had said?
Cortisol.
Cortisol, yes. So you have, but I would just want to mention before we get to cortisol that there's really amazing neonatology researcher at Columbia named Anna Penn, who coined this term neuroplacentology. Have you heard that before, Ryan?
No.
So she coined that in the sense of how important the placenta is for the brain. Neuroplacentology is a field that she is very important in, and she has shown that in her lab that another molecule called allopregnanolone arises from the placenta. Allopregnanolone is in that category of steroid hormones that cortisol is in, and plays a big role in refining brain development, particularly in the cerebellum. So she is obviously interested in her clinical work. She works with prematurely born babies. She's a neonatologist, and is interested in the idea that perhaps providing some of the factors like allopregnanolone might assist brain development. And that's one of the aspects of the work of her lab. But it gets us into steroid hormones in general, which cortisol is one of the steroid hormones. And steroids are very, very important for normal function. If you didn't have steroid hormones, your body really wouldn't function correctly. But they also signal problems sometimes. So cortisol is what's known as the stress hormone. It becomes elevated in levels when a person is undergoing a lot of stress, and it does that so it can signal to the body, oh, you should now do the things that would help you deal with some stress. You should, release the glucose so we have some energy. So if we need to run away, we should do that. It puts your body in a different state so it can respond to stress. Of course, too much stress can also be bad. We all know that. We don't. None of us want to experience too much. And during prenatal life, too much stress on the part of the mother may be a risk. It seems to be a risk factor through a lot of studies for neurodevelopmental disabilities in offspring. That's not to say that the system hasn't attempted to, I'm making it sound like an active player here, but perhaps through evolution, the system seems to have developed so that too much stress hormone can't affect the offspring. There are active functions in the placenta to monitor how much stress hormone, how much cortisol is coming from the mother, and convert it into things that aren't going to cause problems for the fetus. But there are points at which that system is not as good. It doesn't function as well. And also stresses that cause so much of that activation and so much of that cortisol that likely it supersedes, it overcomes those barriers. And that's something my lab studies a lot of, is the impact of stress on the developing offspring brain. And we have found, in fact, that these studies in humans have suggested it's a risk factor for neurodevelopmental disabilities. And we, among other labs, have shown, in fact, that it causes specific developmental delays in the brain. The first one that we had studied was a delay in the migration of cells that have to travel long distances in the brain. And now we're interested in looking at other ways of understanding whether the brain gets delayed and maybe stays in a period of growth for a longer period of time than it should, as perhaps one way of responding to cortisol or other factors that happen in stress. So stress is a complex state and involves inflammation, it involves cortisol, it involves changes in your cardiovascular function, all of which might be part of the reason it is a risk factor.
Okay. Cortisol. It often gets it's a bit of a misnomer, I think, because true, we associate it with stress so much, but naturally it's what wakes us up in the morning. So something is telling me that it's actually a good thing. We just, maybe as modern humans, kind of just don't know what it's supposed to be doing.
I think you're right. Yeah, exactly. I mean, and even at the phenomenological level, we know that having absolutely no stress in your life isn't a good thing. So even if we want to always put it together with stress, it would be bad for it to be gone entirely.
Yeah. And where it's cleaved from in the hypothalamus is very telling. It's very fascinating, I should say. From POMC, the ACTH, the hypothalamus cleaves it, and then from the pituitary, the ACTH, but the ACTH and the CLIP hormone, and then below that in this biosynthesis is a melanocyte. So it's like, whenever we were discussing the leukocytes, I'm thinking about the alpha melanocyte as well because of its role in leukocytes. And these things are kind of like connected, or are we just not doing what we're supposed to be doing with these molecules in these biosynthesis? Because autism just kind of shows up in the 1930s. So what really happened? What's changed?
Oh, that's such a good question. So many people are interested in understanding why we are describing more children now, and maybe even beginning to describe them only 100 years ago, and not just children, people throughout their lifespan having these particular problems. As with anything, I think there's a role for better understanding, leading people to recognize it more, right? That the more we understand something and the more we feel like we have something that we can say about it or do about it, we will tend to notice it more. But there's also the possibility that the rates have also changed. And it's hard to understand exactly why that would be. We know for sure that genetics plays a huge role in autism. So is it something that interacts with genetics? Is it something that's adding to genetics? These are all great questions that I don't think we have all the answers for. And I don't know that much about melanocytes and leukocyte interaction. So I don't know that I can comment on that.
Yeah. That's okay. Sometimes I just get excited.
Yeah, no. That'll be the next podcast.
Yeah. It's always good to have more and more topics coming.
Yeah.
Let's talk about. Well, there's two items here while we're on the placenta in this prenatal time frame. I'll just mention them both. But placenta's role in gene expression and DNA methylation. And then also, if you know anything about cell proliferation, then the differentiation, and then you mentioned migration earlier. You can just.
Sure, absolutely. So placenta, in terms of methylation, well, so I mean, in case your listeners aren't up on methylation, that's a way that the genes that are the same in every single, well, not every single, in most of the cells of my body, I have the same genes, but they're doing very different things in different cells, and they do very different things at different ages as well. And we think that a lot of that regulation for how does the set of genes I have do something different in different scenarios comes from whether they're methylated or other modifications that are similar to methylation. So not to put it all on the back of methylation, but it's not that methylation reflects developmental stage, so that there's like normal changes in methylation that occur, like when we make gametes that are going to produce like an embryo when they join an egg and sperm. There's methylation changes that occur through the division of the earliest cells of an embryo, blastocyst stage. And then there's also methylation that's thought to reflect exposures and sort of keeping almost like a way of the cell of keeping track of things that have happened to it historically. And then there's also, at a completely different level of the role of methylation is in the brain, very rapid changes in methylation, like when we're sitting here talking to each other, are probably part of what's allowing us to build memories of what we're doing. And so that's another place that DNA methylation plays a role. But in the placenta, it is interesting how, just like with any cell, the methylation pattern might be able to tell us something about how the system was functioning, as well as what the system had experienced before. And that's becoming a way for us to possibly look at function of the placenta, but also sort of the history of the placenta, because it's a tissue, as we talked about earlier, that has a role for the period of pregnancy, and then it has nothing, then we're done with it. And but some researchers are starting to collect samples of placenta from lots of different pregnancies so that we could maybe look at methylation, among other things, to understand better, does the methylation of the placenta differ between people who eventually are diagnosed with autism, eventually diagnosed with ADHD, or anything else, like perhaps even understanding other diseases such as obesity, just because the placenta plays a role in so much of, it's intimately involved in all of fetal development when some of these problems may begin. So I'm not sure if there was anything particular about methylation that you had been thinking about, Ryan.
Not anything in particular. I just know that it's pretty common in the literature. So,
Yeah.
it's just, about the only thing I really know about it is it takes a donor from methionine. It's often things in neurobiology have those cycles, and then it gives the donor to folate, and then on from there. So,
yeah, and so you're basically in part pointing out that methylation can't just happen just because because the cell wants to have methylation. It has to have the right cofactors there to actually make the methylation happen. And that can also be a way that you can know if, there's been perhaps a limitation of certain nutrients by virtue of methylation patterns. But something else you said earlier, maybe, we were talking about developmental processes of things like migration and proliferation. And so in placenta, the cell, making that tissue and forming that tissue rapidly does involve those normal processes of cells dividing, proliferating, and cells migrating to where they need to go, including cells that contain particular nutrients that have to locate themselves particularly in certain parts of the placenta so that they can provide the nutrients to the fetus. But I had referred to some processes like proliferation and migration with respect to the fetal brain, because the fetal brain has a very systematic way that it gets put together, and that involves cells being rapidly produced. You have to produce thousands, tens of thousands of cells very quickly. And in fact, we don't produce really, for the most part, any more neurons after the prenatal period, not to any large extent. In very small numbers are there new neurons produced. So we get all of the neurons we're ever going to need to do all the stuff we're doing, talking, the thinking, all this stuff in the prenatal period. And those cells are all born in sort of if you think of the developing brain, this is maybe very, very simplistic, as sort of a donut shape. It has a hole in the middle, and it's sort of a donut. And that hole in the middle is sort of this fluid-filled space, and all the cells that are born right along the edge of that hole, and then they have to migrate away from that edge of the hole into the rest of the donut. And so cell migration is a major phase of brain development that requires a lot of energy because the cells have to move and requires very, very specific guiding signals so the cells know where to move. And some of those things we think were in the models that we looked at. The migration of cells seemed to be the cells weren't getting the right cues on where to go when they were in the presence of stress, maternal stress, I should say. And we actually think that another way of describing another way of using the word stress is in a cellular context is oxidative stress. And for cells, that's not the same as the kind of stress you and I as people experience. It's like a cell experiences an imbalance in the level of like damaging molecules it gets exposed to. And we think that oxidative stress is playing a role in misdirecting some of that migration during prenatal brain development, that might also contribute to neurodevelopmental disorders.
The source of the oxidative stress, is that from the mother, or is that in the womb itself?
Yeah, I think it's getting focused on it originates probably in some of the very specific aspects of maternal physiology that change in certain circumstance. Like we talked about, maternal stress can involve cortisol, it can involve inflammation, and those processes, when directed at particular cells, like the cells of the placenta, the cells of the fetal brain, can actually then secondarily induce oxidative stress. So it sort of originates in the mother, but it's filtered through the placenta and the fetal tissue response to those things that are happening from the mother. We have found in some of our models that in part those effects can be blocked by antioxidants. So people have heard of that probably because we hear about antioxidants in our diet. And antioxidants are molecules that can combat oxidative stress. And when we have provided that in our models where we think there's a high level of oxidative stress occurring and this is causing a disruption of brain development, we've found correction of some of those processes when the mother is given antioxidants to while also experiencing the sort of the risky condition, the stress condition. So
Okay.
so that we do think that it's not at the level of an exact dose or an exact guidance in diet, but we do think that it's good to have a good level of antioxidants in your diet during pregnancy, just because it can help regulate some of the keep in line in the normal range of operations the levels of you know oxidative stress, not allowing it to get too out of control.
Yeah. I've seen some data saying that autism and mitochondria, there's about 80% implicated with mitochondria, but I'm like, I think that's highly misrepresented. I think it's closer probably to 100% or maybe even
Oh,
100%.
yeah. I mean,
Because,
yeah, I don't know about the percentages, but I mean there are some theories that part of the problem is in mitochondrial regulation of providing energy and then cleaning up the byproduct of producing that energy, which is cleaning up the oxidative stress. So,
yeah,
I mean, it is a
or
very interesting idea, and there are some very specific genetic deficits in that process that cause neurodevelopmental disabilities. And so it does show us that for sure that problem can result in a problem like autism. But I don't know the extent of it across all different people who have autism.
mm, yeah, the mitochondria are it's a bit of a buzzword right now, but it's for a good reason, I think.
So
Um,
important. So
yeah,
important.
yeah. There's so many of them though, so it's kind of hard to dial in. It's easy to look at the role of one, but like the cell nucleus, there's so many. How
Yeah.
many does it take? How many abnormal mitochondria in a cell nucleus does it take for the cell to kind of underperform? Is
It's a
I don't
very
know.
good, very good question. And it really probably depends on what the cell's trying to do,
Yeah,
the
yeah.
stage of development, all the other things going on at the same time. That's why biology is so interesting, yet so,
Mm.
so confoundedly complex.
Yeah. Whenever you mentioned oxygen earlier, I instantly went to mitochondria. Uh,
Hmm.
Dr. Richard Fry was on, and he said just nonchalantly, he said the whole reason we breathe is so the oxygen can function into cytochrome C oxidase. So that's that's the whole
I
reason we
love
breathe.
that. I love that
Yeah.
observation. That's a great one. Yeah.
I would have never thought, well, we breathe, so for that reason in the
Yeah.
mitochondria. But with even what we know about melatonin being produced in the mitochondria during the day, and then that maybe being, if we were to subordinate them, melatonin being our number one antioxidant. Of course, there are other crucial ones, but is something like that the daytime environment, maybe even like us being removed from the sunlight now, and because of the electrons, are, are we deficient on electrons? Um,
Hmm. Oh, so you're thinking about like changes in modern life as we
yeah,
having and the rates of autism going up and less sun exposure. That's interesting. That's interesting. I would say that like there's so many things contributing to um changes in the provision of certain molecules. I modern diet, major shift, right? Like we eat so differently than we did 100 years ago. So that's another one. But yeah, that's a very interesting hypothesis.
I'm hitched on it though, Dr. Stevens. So I just have to caution you that autism predates all of these other environmental toxins that are discovered now. I'm not saying that they're not important, but the artificial light predates autism. And then light is, if we think about quantum physics, light is electrons. And it doesn't have, it hits matter, and then, it just causes so much energy to come about that. And we've neglected ourselves. And I think maybe that's why autism shows up roughly two
Mm,
generations after artificial light.
interesting. There is a field of biology called quantum biology. I don't know if you've heard of
Yeah,
that, where
yeah.
people are trying to understand the role of quantum particles in the processes of cells, which is really it starts to blow my mind even more than most biology does.
Yeah.
So, yeah, it's a
'Cause
lot
This
to
is
think
at
about.
the this is as far upstream in the cell that as you can go is
Right.
quantum biology,
Yes.
because um, it's what drives biochemistry. Biochemistry doesn't work without the quantum physics, the signals from the environment is um, is what I'm taught by
Yeah.
these.
Yeah.
But um,
Oh, big ideas. Wow. I
yeah, and then
I can't
so
comment, I don't think.
autism it originates in the 1930s, and the rates also follow as us removing ourselves more from the sun and introducing artificial light and the types of artificial light, because light has a wavelength.
I wonder how, with these disorders that are so modern, that have modern definitions, I wonder if we how we would ever be able to determine if people did have these problems before.
Yeah,
You do see characters in literature that was written hundreds of years ago that appear to have some of the issues of modernly defined neurodevelopmental problems. I particularly think about ADHD in that way.
yeah.
And then the historical record, there's quite a bit of reference to people who struggle with attention and
Mhm.
control of movement and things like that. But getting a rate would be so hard, a real rate. So it's always going to be a bit of a mystery to know how much it was, happening 100 years ago. And then, the cultural response to it too is like maybe people were even, there's more stigma maybe in the past.
Mm.
So maybe people didn't even have a chance to know people who might have had autism. I don't know. These are big
Yeah,
ideas to think about.
ADHD goes back a couple hundred years, 17th century maybe, and our modern environment doesn't do it any favors.
Oh,
Sit down,
that's true.
still.
All the distractions are pretty
Yeah,
tough. Yep.
yeah.
Yep. Yeah.
So,
Yeah, for sure.
boy, we haven't even talked about the dorsal striatum,
I
but
know we haven't, but we've
but
already been talking a long time.
yeah, that's okay. But I do have a couple of questions for you before we wrap up.
Sure.
I love the dorsal striatum so much, but I I realize we're not going to get into it. But I am very curious about what are you recently most excited about? Be personal or professional, your research.
Yeah. Well, since you've alluded to the dorsal striatum, I will say we have some really interesting data. So one of the um, one of the things to think about if we're trying to understand how prenatal events in brain development influence the risk for neurodevelopmental disorders, one of the major factors that happens is determining the right size of a structure and the right cellular population that goes into that structure. And so we've been interested in that from the sake of the dorsal striatum. Many people have focused on that question from the standpoint of the cerebral cortex. So there's been a huge range of literature going back 20 years on what regulates the right number of cells populating the
Mm.
cortex. And people have studied genetic factors and they've studied environmental factors and have emphasized the role of these wonderful cells, the microglia, in regulating those processes, but not as much has really been done about how do we regulate the right size of the dorsal striatum. And because of evidence in the literature that autism seems to have enlarged dorsal striatum from the earliest time point that people look at brain imaging studies of people with autism, we wanted to understand, what if we try to manipulate the size and the cell population that goes into building the striatum? And so we have really new data that we're just going to present this coming week and is still really being collected, showing that if we use a modulator of cell proliferation in mice while their brains are developing in utero, we actually seem to be able to increase the size of the striatum and disrupt some of the kinds of problems that, or some of the kind of behaviors that are also disrupted in neurodevelopmental disorders, including things like habit learning or procedural learning, which the striatum plays a huge role in, is allowing us to learn how to do things in a very regulated, patterned way, just so we don't have to think about it every single time we do something. So we have new data suggesting that when we manipulate this and grow the striatum beyond the point where it normally will grow, that we see this disruption of these procedural learning behaviors in the animals after they grow up. And that they're the one thing we really want to understand is what happens when we increase that growth early? Does it stay increased? Does it actually get even larger over time? Because one of the other phenomena in autism is that the dorsal striatum doesn't just begin larger, it actually seems to grow faster through childhood. And so we want to look at this model, if we really have been successful in creating it, and understand how it tracks over time and how it changes with development, because we think that might help us understand some of the places where you can't change the initial events that occur prenatally because you don't yet know that you have the issue, but could we find out how it tracks with time, and then find ways to, if a person is having a big problem because of some of the symptoms, find ways to modify the striatal cells, the striatal structure in ways that would be beneficial. So that's really an exciting thing for us right now.
That is exciting. Do you know if it's the larger size is increased amounts of dendrites and axons, and even the morphology of it?
That's one of the questions we want to find out. So thus far, we've been focusing on creating more of the cells that build the structure, because we see those differences in people with autism, and it seems to suggest that it's an early fundamental problem like the cell population, but it might lead to what you're saying also, that you have alterations in dendrite and synaptic structure. And that would be something we have to explore after after the next stage of the study. It's coming right up. So
Great.
yeah, hopefully we'll be able to answer that question soon.
Yeah, I'm looking forward to that. It's
Great.
it kind of makes sense, because it's then those areas become preferred, because our central nervous system just wants to respond. It doesn't like to work. It just wants to respond based off of what it knows.
Right. Yep. Yeah. And I mean, that would suggest, we know that even if we don't necessarily see increased structure in people with autism, we know that striatal connectivity to the sensory cortex is also more than in people who don't have autism. So we have like the cells, not only maybe in some people there's more of them, but maybe in other people it's not that there's more cells, it's that they're just more active. And understanding why that would be another goal. Maybe that's, when we see these things happen in people, we don't know for sure if it's like that was part of the thing that caused the problem, or is that actually what the person is doing to try to deal with the problem?
Yeah.
So, that's still up for interpretation, but certainly worth exploring.
Yeah, that's awesome. Well, Dr. Stevens, I just loved our conversation, and I thank you for your time. I'm looking forward to the data that you just explained, and I'll be looking out for it.
Oh, fantastic. Well, I'll keep you updated for sure.
Yeah, please do.
That would be great. That would be great. Well, thank
Well,
you so much for all your interest in the placenta. I'm really hopeful that the big take home point is I'm just hopeful that we can do the right thing to support healthy brain development as much as possible, as early as possible. And that really all starts by, making sure we know what goes into a healthy brain in the earliest time points. So
yeah,
that's my goal.
that's awesome. That's my number two on my list. Number one is cause, and number two is identification and early biomarkers, because
Exactly.
it's undisputed that the earlier the better. Yeah.
Earlier the better.
Well,
Great.
thank you. Have a great weekend, and thank you so much.
Yeah, take care.
If you are listening to the podcast or listening to the episode, please feel free to leave a review or ratings. In podcasting, review and ratings are crucial, and I very much appreciate your feedback. You can contact me on X at RPS47586 or click on the hop link so you can have links to all the show platforms and contact information. You can email me info.fromthespectrum@gmail.com. And thank you for listening to From the Spectrum podcast.